that's what the connection looks like. We have our deoxyribose sugar. We have sugar, phosphate, and so on. Each one of these bonds, here to here, is an ester bond. So there's an ester bond there, and an ester bond there, which makes this a phosphodieester bond. So the bond that links the sugars in the chain is a diester, two single ester bonds. Louder.
On the adenine, you have an OH, but it's supposed to be O or an H.
Thank you. Yes?
That's what I was going to say.
So this is a phosphodiester bond. We have a phosphate in the middle, sugar, phosphate, sugar. So we can represent the phosphodiester bond as carbon, oxygen, phosphorus, oxygen, carbon. So this would be the 5', 3', or 3', 5', depending on how you look at it. So that's the linkage between individual monomer units in the double helix. Any questions? Anyone ask questions on this linkage?
Third carbon on adenine. Are you missing a note down on adenine? Third carbon. Connected to an oxygen. Are you missing a hydrogen?
No, it can't.
You can't do that, right?
You can't be there. You can't. So once you make the ester bond, we'll show that in replication. That's a condensation. You're taking off H2O. So the H is gone. No, this is not OH here. These two oxygen valence of 2 are just exactly the way I've drawn them. Anybody else? Any questions? The phosphodiester bond is the bond that links the monomer subunits to make a polymer DNA chain. Anybody else? Okay, let's do a very simple in-class cooperative learning exercise. Make sure that everybody is on the same page.
Thank you.
Thank you.
So the first one, you don't have a hippity. You do. Chargas law. Chargas, yeah. Percent of C plus D. Percent of A plus G equals the percent of C
If
less.
it's 50% of basis in the material, what is the
So
T and A go together.
No, T.
G and
It's
C go
because they're
together.
it's pyrimidines equal the percent of pure pyrines together. So basically, you have A plus G equals the percent of C plus T. So A equals T. So T is also A equals 15%.
Talk about training class, so I'm prepared. Okay, sharp law equals percent of pyrene. So, T is equal to A percent, and you know that then therefore T and A will equal 50. So that equals 35.
45. 45. Wait, no, 35. 35 plus 35 is 7.
You're making me second guess myself.
No, you got it right. You got it right, yo. Don't second guess yourself, especially when I'm telling you things.
No, it would be 30%. 15 plus 15 is 30.
15 plus 13 is 30, minus 100. 100 minus 30 is 70. 70 is 35 plus 35, right?
I would go more on the
35. It's 45, 45 is 90, right?
yes.
So you do 15 plus 15 is 30, 100 minus 30 is 70, and then 70 divided by 2 is 35. Yep. Not 45.
Is this asking us all together? Or is it individually?
So for, if you know that that's that much for GC, how much is for G and how much is for C?
Well, you wouldn't know. Is it the same? Or is
They're
it,
the same, so I mean,
was
let's
it
say,
half?
yeah, half of that percent.
Okay, so 24. Like, why do you know it's half?
Because you know that G and C, exactly, you know that G and C link together. So there has to be the same amount of G. So together they're 48.
You get 26 for A and B. Yeah. And 24, 24. I like Chargas. I want a whole test like Chargas. And then not have a calculator and list one of them up. Are you getting it? Yes. The pyrene equals the pyrene.
But, so, if factor of A is 48, GC would percent of A.
They're not all equal, but the
So, okay,
T's are
so
equal to the
48
Okay,
with A divided by
think of it like this. For every A, there has to be a T on the other side.
24, right? So 100 minus 48 is what is it? And 52? What's 52 divided by 2? 6, so that's A and T time. So G
Let's just.
Thank you. If a virus has does this make sense?
It does not make sense.
It does not make sense because A and T does not equal itself and G and C don't equal each other.
Problem set 7 is one of my favorite problem sets so far.
I said, you're so energetic. I wish I had half the energy you
I did a
have.
5-hour energy yesterday.
Oh my god. So,
I was like, really?
so like, why?
I had no windows, but like,
It just goes down like, it goes down with like 100 decibels.
Oh my god,
Yes. Maybe obscure.
I could, when I was writing it down, no, I knew what it was. Yeah, I said, what kind of wording I did not, I got like, I put down, I got that one. Basically, like that, when I was like, when I was sharing my PowerPoint, I didn't even spend time with that. But it should like,
Yeah, it does equal A plus G.
Yep.
no, there is B, so it's
that's just like,
because they're single stranded and I can like very good.
You would know from this that it's single stranded.
Oh, okay.
Because if it's double stranded, then it would have to say. Go ahead.
What if it was like AIDS? Where it's an RNA virus?
What?
What if it was like AIDS? Would Chargaff's rule still work? Because it's an RNA virus, so you'd have to use instead
Well,
of
even with RNA, if it's double-stranded RNA, then it would work. The thing here is, okay, if we're talking about a viral,
just a
genetic
general
material,
one. Okay.
so it could be RNA or DNA. Because there is P involved, you know it's DNA already. Does it have to be double-stranded though? No. So then Chargaff's rule doesn't need to apply. Because if you just have one, it doesn't have to match up with anything on the other side. So that's basically your explanation, is it is single-stranded.
Oh, so that's
Because it's a virus, right?
so
And
because it can be single. Okay.
It-
So
it-
actually, so Chargaff's rule, it's
you know that it's single-stranded here because it doesn't
it
have.
doesn't add up.
If it does have equal amounts, you could say it could be single-stranded or double-stranded because it could be a single strand with equal amounts.
So we're actually saying that it's still a no that Chargaff's rule doesn't work. But
It doesn't work, but the reason.
But the
for
reason
it,
why is because it's a single strand. Okay.
Exactly.
So in this case, it would work. It would have applied it because it's a virus. It's not from the virus. Yeah. Are all viruses single stranded? They all are? Okay, that's why. They're RNA. They're RNA. They're RNA and RNA. Okay. Okay, that makes sense.
If it injects one,
out over the weekend. It'll tell you if you're right or wrong. When you hear the answer, you say, oh my God, that was so simple. So just keep thinking about it. So now, you can move on. Now that you understand DNA structure, we're going to move up the ladder of complexity, and now we're going to look at chromosomes. And we're going to break this up, because we have to break it up and look separately at bacteria and eukaryotic chromosomes. We'll go through this, and ultimately, we are going to end up building a model human chromosome. One of the constituent parts of a model human chromosome. So, in general, bacterial chromosomes are small, eukaryotic chromosomes are large. Typically, bacteria have one chromosome, but so all of these are just typical features. Typically, eukaryotes have multiple large chromosomes. What we're going to look at now is something called DNA sequence organization. So, just in general, it's very hard to explain. It will become really clear once we start to go through it. What I mean by DNA sequence organization, the emphasis is on the organization part. What that means is we are not going to look at DNA sequence. That will come later when we look at genes. What we're interested in here is how chromosomes are put together. What you're going to see is that chromosomes are put together in blocks. So we're going to look at DNA sequence organization, let me drop that down, in two different ways. So the first way, we're going to look at GC distribution. If you remember from Chargaff's work, what he described is the base composition of different species. And we had all those things that were equal. So A equals T, G equals C, purines equals pyrimidines. And the one thing that differed, actually two, the one thing that differed is the GC content. As GC goes up, according to his rules, AT goes down. So GC plus AT equals 100%. So the question now becomes, what is the distribution of GC base pairs in a chromosome? So we can take a model. We're going to start with bacteria. So we have a circular chromosome. They're not always circular, but almost always. And let's say that we have 50% GC. We can break this chromosome up into fragments and ask, what is the GC content of each fragment? And so this is a model of DNA sequence organization where every fragment that you make in a chromosome has the same GC content. Therefore, we describe the distribution as a uniform distribution around the chromosome. So, this is model A. Model B, we take another chromosome, another bacterial species, break it up, and ask what is the GC distribution here. 50, 45, 55, 35, 30, 25, 20, 15, 10, 35, 45. Model B has a completely different DNA sequence organization with regard to the distribution of GC base pairs. And so this second model is not a uniform distribution. So to answer the question of which model an individual species has, we need a technique or a tool to enable us to distinguish DNA fragments of different GC content. And that tool is cesium chloride gradient centrifugation. So cesium chloride is just a salt. It's in the same family as sodium chloride, and it's ideal for this kind of an analysis. So gradient means that we're going to take a test tube and fill it with a solution, a salt solution of cesium chloride, such that we have a low concentration on top and a high concentration on the bottom. And if we were to drip this out into a series of test tubes and fractionate this, and then graph cesium chloride concentration, what we would see is a uniform distribution from top, a uniform gradient distribution from top to bottom. Now the reason that we're doing this, we can make this, it's actually very easy to make. Centrifugation means that we are going to put it in a machine and spin this around at a really high speed. We can go 40, 50,000 revolutions per minute, which imparts a huge gravitational field on the material in the solution. We can get hundreds of thousands times gravity. We're sitting spinning at 1 G. We can increase that by spinning it super fast to hundreds of thousands times G. And we can get that DNA. I'm not going to tell you how now. I'm going to come to the mechanism, maybe a few weeks. But for now, all you need to know, the important thing, is that we can take these fragments, and the fragments will separate by GC content. So each fragment that has a different GC content will be at a different position in the gradient. So we have our two models, and we can make predictions of what we expect to see based on the experimental protocol. The experimental protocol will separate each piece of DNA that has a different GC content into a different band in the solution.
So if it was a straight line, it'd be uniform.
So, right, so the bands will look like a straight line sitting, floating at a position in the solution. And I don't want to get into why, we'll do that pretty soon. Does everybody see what we're going to do? We're going to separate out DNAs of different GC content. So for this uniform distribution, what is your prediction for what the DNA will look like in this test tube? So how many bands? Everybody agree? I only heard 7 people. Does everybody agree that model A will give one single band which will graph out like that on the graph? Because every DNA fragment in model A has the same GC content, cesium chloride, gradient centrifugation separates fragments of different GC content and puts them in different bands. Because every fragment has the same GC content, we see one band, and when we fractionate this, we get a single peak on the gradient. Everybody see that all right? Yes.
I'm not sure I understand what's the straight line on.
Oh, straight line is cesium chloride concentration, just showing you it's a linear gradient inside. Actually, it's not quite as linear as the ends curve. Any questions on the prediction you have made that if the bacteria has a uniform distribution, this is the result that you will see? Okay. What is your prediction for model B?
Well, wavy.
Multiple bands, a series of bands. Does everybody agree with that? Is everybody participating?
Yep.
Okay. So those are the 2 predictions. When you actually do the experiment, you fractionate the DNA. First you extract chromosomes, and you do it for hundreds of millions of bacteria because they're small. Break them up randomly. It's random fractionation, but you can sort of control the size. And what you find is model A. For every single bacteria that I have ever seen analyzed, model A is the mechanism.
Is that because they're combining all together, so it's going to mix? Because you're using essentially pull apart and go into their respective groups either way, right? So they wouldn't be separated like you would in the model.
I guess I don't understand what you're saying. You're talking about double-stranded DNA.
Yeah.
They stay double-stranded. You're not going to melt the DNA. That's a separate issue. So,
Well, I guess what I was saying is based on the model of the actual layout of it, you're not going to have that distribution because everything's going to combine together. It's going to be broken down chemicals, so you're still going to have that.
so we're not breaking anything down chemically. What we're doing is making fragments of DNA. And the fragments are either all the same, or each fragment has a different GC content. So again, I don't understand what you're asking.
Okay, I'm going to just forget what I just said and listen to you say.
So are you sure you understand what I'm saying?
I understand what you're saying, I just got a concept that was off.
So the only question is, if this were the model, this would be the data. But it's not. this is the only data you ever see. All right? So now, first of all, we can get rid of this. So that's bacteria.
But it's still 50% GC content, right?
Yes.
B? So of
It's
course
still
it's going to
50.
be the same as A, right?
DNA,
No, because you could have 50% for someone else and you have random assortment for each side.
You're
Like you could
breaking it
have,
all up and overall it's 50%.
yes, but he's saying that each individual second could have other values. But the whole chromosome is 50% GC.
Yeah, but you're taking the whole chromosome, breaking it all up.
Yeah.
So it's 50%.
Yeah.
I didn't understand why he was saying.
chromosomes in bacteria are DNA. The chromosomes in eukaryote are chromatin, which is DNA plus protein. So the chromosome itself is quite different. Any last questions? Now we're going to have to do the same thing for a eukaryotic chromosome, for your chromosome. Any questions on what we've done here? All right. So now, actually, I don't even need that. You guys. So when we break the chromosome up, we still have the exact same two options. Either the DNA is all the same, or different DNA fragments have different GC content. So we're going to put it in a cesium chloride gradient. We're going to spin it in a centrifuge. And what I'm going to show you is the data. So, compared to the bacterial data, what is your interpretation of what your chromosome DNA sequence organization or GC content is?
Non-uniform?
It's not uniform. It is not uniform. Everybody agree with that? So what we have is a very different picture from the bacterial chromosome picture. Now we can label this because we have all different kinds of peaks on here. This is called MB for main band. That's obvious. There is typically in all eukaryotes one single major main band. Then we have all these little peaks in here. These are satellite peaks. And so we talk about main band DNA and satellite DNA. Every species is going to have a different picture. But that is, the picture will differ in terms of what you're looking at. What is the percentage of main band? How many satellites? I've gone 6. I just completely made this up on the spot. Different species will have different number of satellite peaks. The percentage of main band to satellites will be different in different species. The distribution of satellites. So I have 2, so this is heavier and lighter. Heavier means more GC. So the distribution of satellites is different in different species. And so this is another one of these biochemical descriptions of a species. You can describe it in terms of how many legs, how many wings, and so on. You can describe a species in terms of its GC content. You can describe a species in terms of its karyotype. And now we're focusing in more on the biochemistry. So different species will each have a different graph, different proportions on that graph. Now, that's for the species. Everybody in this room has exactly the same pattern for their DNA. The Y chromosome is not big enough to make a difference in this mechanism by which we've separated out the DNA. Any questions so far? Satellite DNA has a very simple definition. It is non-main band. Period. There is no functionality attached to satellite DNA. We're going to talk about features of satellite DNA next week, what sorts of things we see in satellite DNA, what sorts of things we see in main band, but there is no functionality. So satellite DNA simply means that all the DNA fragments, so we have one big heavy main band and a bunch of satellites. It just says that the DNA fragment that ended up in this band down here has a different GC content than main band. So there's no function attached to it. Does everybody know what silk is? Silk is actually a protein, which means there's a silk gene in that moth. And the way it works is that it's a very unusual protein. That's a very unusual gene. That gene has a very high GC content. So if you were to superimpose a test for the silk gene, what you would see is something that looks like that. You don't even see it because it's only 2 genes, it's diploid, out of all the DNA. But if you were to assay it for specifically, it would be way down at the bottom because it is so GC rich. So, by 2 different ways, by this first way, what we see is that bacterial chromosomes are simpler. The DNA sequence organization is simpler than bacteria. Any questions on that? Yes?
What's the y-axis? Axis on that
Here? Here? It's the same thing. So it's the percent GC for this, and it's the CC concentration for that one. So it's done exactly the same way. Yeah.
So that graph basically says that most of your DNA has whatever the main band coefficient of CG
So
is.
most of your DNA is main band. Now that's not true for all organisms. Plants, for example, have a much higher percentage of satellite DNA than animals. Not all plants, but especially higher plants. And so again, each species will have a different picture here. The percentage of main band of satellites, the number of satellites is going to be unique. So it's an identifier for the species. Anybody else? Yeah. Well, so AT is just the reverse of GC. So if it's 70% GC, you know it's 30% AT. But somehow from Chargaff's papers, where he said everything is the same, A equals T, G equals all that, he said GC content differs. And that's what's stuck in the literature. But you know, as GC differs, so does AT inversely. Yes?
So did he just arbitrarily decide to categorize organisms as GC?
Apparently, whether he flipped the coin, I honestly don't know. But GC got fixed in the literature very, very early, after 1953. Anybody else? Yes?
Is there a pattern for like fragmenting the DNA? Like, how do they?
So no, there's no pattern. It's totally random. In fact, DNA is so fragile. If we extract DNA from a bacteria or a liver from you, we get the DNA out, it's already broken. And then to break it down, typically about 1,000 bases, about a kb size, that was conjugated. So it's all completely random. You have hundreds of copies of the same chromosome. Each one is broken up differently.
So then would these tests, they could be like a little bit variable?
No, that's the whole point. It's not variable. This is a method of looking at the chromosomes where the individual chromosome doesn't matter. It's the big picture that we're trying to get now. We're going to zoom in next week and look at the detail. Right now, this is the big picture for what chromosomes look like.
So the GC content is not 50% in this one?
Yours is not. It's close.
Yeah.
Most organisms are fairly close, and for real scientific work, you need to go out one decimal. But overall, it varies from about over 35% to about 65% or more. So there's a big variation in different species.
Can you tell the percentage from this graph, or is that not?
No, there's too much data there. No, you'd have to do a Chargaff experiment if you want to know the actual percent. Yes?
On the test tube, is it high, low concentration of cesium or GC content?
So this graph is percent GC, but also percent cesium. And the cesium low concentration of cesium. So the DNA is separated based on the concentration of cesium. And again, we'll get into that mechanism probably in a couple of weeks.
So high cesium is low CG?
Right. So low cesium is low GC. That's
High cesium is, because it's,
high GC is down here. The bottom is high.
So shouldn't the line go that way?
Right. So this is low GC.
yeah, but it's high cesium, right?
No, it's got to be low cesium on top.
Yeah, okay.
It's low cesium, yeah, like backwards.
All right.
So then what does that
The
line
bottom is
say?
like the part of
That's
the
percent
tube.
GC?
Yeah, because I know the bottom part is high cesium.
Yeah,
So that means high GC, the lower.
yeah.
Okay.
Because you spin it, so all the GC goes to like the bottom of the tube.
Yep.